All right. So the lymphatic system really does lend itself easily to go right into the immune system. So does our discussion of blood. That's why this is all together kind of in the same unit. And in our immune system, we're going to see, much like the lymphatic system is filtering material, it is moving this debris and pathogens to areas like lymph nodes, the thymus, the spleen, such that these foreign material pathogens can be dealt with. And now we're expanding it. We're talking about bacteria, viruses, parasites. There's a lot of things. And there are a lot of different organs that are going to play roles in this immune system, not just our secondary lymphoid vessels, our diffuse lymphatic tissue. But first, we want to talk about what are pathogens? Pathogens cause pathology. Pathology generating materials. That's where the word pathogen comes from. So these are infectious agents that can kill you, can cause a lot of damage, and certainly they can make you feel like crap. And we group these into 6 sort of major categories. I don't know that these are necessarily all inclusive, but most pathogens can fall into one of these 6 categories, and we're going to take these one at a time. This one is one you probably have least heard about, if ever have heard about it. These are called prions. Prions are infectious proteins, and basically they're proteins that have misfolded. You remember our proteins? What is the primary structure of a protein? The amino acid sequence. What is the secondary? That's going to be your alpha helix or your beta sheets. The tertiary is how it's completely folded into a globular protein. In this one, we're supposed to have 4 helices, but in this one, we only got the 2 helices and a beta sheet. So this one has a aberrant, error-filled tertiary structure, and that's going to clog up the works. It's not going to be functional, or its functions are going to be very different, and it can cause major problems. Here's a person with a normal brain. Here is the accumulation of prions in the brain of an individual. And yeah, it's going to cause a lot of problems, and it can even, depending on where this accumulation happens, it can be fatal. So these do happen, and I think you can appreciate these are going to happen in more elderly individuals than in young people. But if you are susceptible to the prion gene, the prion mutation, it can hit at any time in your life. And again, it's just all about misfolding of normal proteins and how they accumulate in, specifically in your central nervous system tissue. Viruses. Unfortunately, we're pretty familiar with viruses, aren't we? Especially after 2020. Yeah, viruses are not cells. Viruses are not living. They do a lot of things, but by our biological definition, they're not living. They're simply pieces of DNA or RNA, you typically won't find both in the same virus, that are encapsulated by protein, an envelope, that then has proteins on top of that. Now, we're familiar with the proteins of coronavirus. We know coronavirus, all right? COVID. Coronavirus has those spike proteins. That's what they were used in those RNA vaccines to give you the RNA so your cells would make the coronavirus spike proteins, and your immune system would say, hey, that's not supposed to be here. But when we look at viruses, common cold's a virus, somebody just sneezed, that's probably you. We hope you don't have Ebola. Chickenpox? Anybody had chickenpox? Are you serious? Have you had chickenpox? I'm the only one that's had chickenpox?
Well, my mom told me
Oh, here we go with old stories. Okay. Huh?
My mom told me that they used to purposely infect or try to purposely expose.
Yeah, they do.
So-
yeah, if you got like if you have a kid and then you have multiple kids, one has chickenpox, you get them all in the room, get it over with, and done.
The antibodies, but then they have shingles
Because you can only get
virus.
once.
We didn't have the chickenpox vaccine. That's why y'all have not had chickenpox. And we'll talk about passive and active immunity vaccines next week. But I was in the first grade when I had chickenpox. My mother had a chickenpox party. And other parents brought their kids over who had not had the chickenpox.
Yeah.
Because,
You know what, as an adult.
right, it's horrible, it's bad enough as a kid, but it's not as dangerous. I have heard, I have not looked this up, I have heard that today, you can, now don't go looking up on Amazon or anything right now, okay, wait till after class. But I have heard you can order lollipops that people that have had chickenpox have like licked. And you can give those to your kids. I've not looked it up. I don't know it's true. It's on the internet, it's gotta be true, right? Yeah, but people are buying this knowing what they're buying. I don't know. I don't know. Somebody look it up and tell me if it's true. Maybe I don't want to know. All right. So if viruses are not cells, that means they have to have help in their replication cycle. So they're considered obligate. They got to have help intracellular, meaning they get inside the cell, parasite. Viruses land on, attach to, infect the cell with their own DNA and RNA. And that material, that nucleic acid code, takes over the function of that entire cell, and it turns that cell into a viral factory until so much virus is made inside, the virus erupts, killing the cell, and the viruses go off to do other nasty things. That is the virus.
Did you hear?
No.
You can't order them, they're illegal.
Oh, you can't?
So viruses are parasitic?
Viruses are considered obligate intracellular parasites. Yeah. All right, bacteria. I think we've all had plenty of experience with bacteria. Bacteria are single-celled prokaryotes. What is prokaryote? No nucleus. They have DNA, but they don't have any membrane-bound compartment that stores the DNA. So there's no nuclear membrane. Now, most of our bacteria are harmless, and when we get to the innate immune system, your skin is part of your innate immune system that protects you from pathogens, because you have bacteria on the surface of your skin that protects you from bad bacteria. So that's a good thing. You actually have bacteria living in your intestinal tract that are good bacteria until they get in the wrong place or get out of control.
That's what happens with the staph infection, right? Staph's usually on your skin.
This is H. pylori.
And not in your intestine, I mean on your skin.
Oh yeah, staph. If that gets loose, then you're in trouble. So most are harmless. But we do have what are considered virulent bacteria, and these can cause damage because they can release toxins into your body, and you don't want any part of those. See, there's staph that you were talking about, Garrett, Lyme disease, syphilis, strep throat. Strep throat's going around. Just be careful. I've heard about that. Tuberculosis, that's what you get your skin test for, tuberculosis. And then also the disease chlamydia is also from bacteria. But, okay, now it's going to start getting kind of... Now here's the thing. As a college student, this is going to gross you out. As a parent, you are immune from grossness. You have to be immune from grossness as a parent, because you're going to be in situations where you just can't. You just got to do it. You just got to do it. And so in the case of a fungus, these are eukaryotic cells, and eukaryotic are opposite of prokaryotic, so eukaryotic have a nucleus. And so we're talking about a fungus, but molds and yeast also are in this. Athlete's foot ringworm. These are all parts of this. And there's thrush that babies get in their mouths. So these organisms release digestive enzymes that can begin to irritate and cause damage because their digestion is done outside of their bodies. They just absorb it. And it really is nasty on the skin. It's not fun. It itches, and yeah. And most of this is going to happen with mucosal linings. And so you definitely get them in this end, but you can also get them in the other end. So again, that's where you have your mucosal linings that are warm, that are moist. That's where you can get those. We'll pick up with protozoans on Tuesday. Okay, we got a lot of slides to do, but I don't think it's going to take us a lot of time between today and Thursday because some of the slides duplicate, reinforce each other. So let's finish up this immune system portion of our bad stuff, the bad things that we're dealing with. We left off with protozoans, and protozoans are eukaryotic. And what did we say that means? Have a nucleus. The EU means good. Karyon is nucleus. So these are good nucleus. They have that nuclear membrane. Now, without a cell wall is significant because these are single-cell organisms, and bacteria have basically a cell wall. But these eukaryotic cells do not, and we put them in the realm of the protozoans. And some examples that you may encounter in your clinics are the malaria protozoan, and you can see that here in these red blood cells. That's where they infect the red blood cells and they reproduce. So it's a bloodborne disease. Now, what population of people in the world basically have a resistance to malaria? Patients with sickle cell anemia are resistant to the malaria protozoan, the parasite. That perpetuated in populations because in Africa, people would die sooner from the parasite than from sickle cell anemia. But now that gene, the sickle cell gene, is so prevalent in populations around the world, in areas where malaria is not really an issue, it is the sickle cell gene that then shortens the life expectancy. So depending on where you are in the world, sickle cell is a good thing if there's a lot of malaria, but if there's not, then it's a bad thing.
Is that an example of natural selection?
It could be thought so, but then when you change the natural pressures, that being the malaria parasite, now you're at a disadvantage. Okay. So it doesn't select itself.
There's a whole thing in bio too.
Yep. Giardia, that's another one, and nobody wants that. It's an intestinal parasite, and do I need to say anything else? Intestinal? Yeah. Montezuma's revenge kind of thing. Now again, these parasites, intracellular, like we see here with the malaria. You can also have extracellular here with the Giardia. Either way, you don't want them, and your immune system is trying to protect you from these protozoans. Yeah, okay, if y'all are going to be nurses, this is going to be the least gross thing you're ever going to see. So these are multicellular parasites, and they're going to live off the host, and a lot of these are going to be internal in your GI tract. And what we see at the top, these are tapeworms that came out of the GI tract of patients, and these are roundworms. And this one has got someone's hand in here for scale. That's not a small worm. And tapeworms can be feet and feet long. So these are some bad things you're going to encounter, and these are just a few of the things that your immune system and your immune cells are trying to protect you against day after day after day. So let's move into our immune cells. This probably looks a little familiar, just a little different. When we talked about blood and our blood cells, we had our stem cell, and then we had our two lineages, right? We've got our myeloid, which is practically everything, and then the lymphoid lineages, which are our lymphocytes. Well, as we're looking at the immune system, we can basically right off the bat eliminate the erythrocyte and the platelet. They don't function in the immune system to any appreciable degree. So we're going to look at these different cell types that function here, and some of them are actually the same. They just changed the name, and we'll see how that works. And we're going to begin to group these, especially these myeloid cells. We're going to group them into subcategories. And the first subcategory we're going to call granulocytes. Now, cytes means cell. Granulo, what do you think that means? Granular. So when light microscopists early in histology looked at these cells, they were like, these things are filled with granules. Now, mast cells is going to be a different name for a basophil. I wish they had extended that on down for this illustration. But all of our other leukocytes don't have these specific granules. And what does the prefix a or an mean in front of a word? Not, without. So if these are granulocytes, guess what everybody else is? A granulocyte. So those are the two groups that we're going to break this out to in just a bit. Yes, sir?
Are the hematopoietic stem cells the same thing as multipotent cells?
Yes, from what we had seen before. So what we see with our granulocytes, these are going to be myeloid lineage cells. And we've already mentioned they have these very visible granules that are going to be differing in size and differing in color, how they stain with histological stains. That's how we're going to name them. And these specific granules are going to be filled some with enzymes. Some are going to be filled with other chemicals that may lead to inflammation, allergic reactions that can cause asthma in a clinical setting. And others are going to have chemicals that can attract other immune cells. We're going to call those chemokines or cytokines. So these specific granules have some really physiological good things that are happening. You as a patient might not experience that because you could have fever, you could be stuffy, you could have a runny nose, all these inconveniences, but that's part of the immune process. And so we're going to give details about these three types of granulocytes first before we get to the A granulocytes that will include the lymphocytes. So neutrophils. The "phil" part of these names comes from philic or loving. And so when the histologists looked at the stain of these, they said, you know, these granules are pretty small, and their color is about the same color as your red blood cells. They're kind of neutral. So someone said, oh, well, they're really neutral loving. They don't really prefer a red stain or a blue stain, so we'll just call them neutrophils. The other thing that you'll notice about these neutrophils, these granules, and I think you can see them in the cytoplasm here, these granules are not going to be so large that you can't see this very, very weird looking nucleus. Now, this is a single nucleus that you can see it's connected by these chromatin strands. So these are multi-lobed nuclei. And in fact, the older the neutrophil, the more lobes there will be to the nucleus. So you can see all sorts of patterns and shapes and number of lobes. And so another name for neutrophils is polymorphonuclear. We got nuclear, right? The nucleus. Poly, many. Morpho, shape. So another name for neutrophil is a PMN, a polymorphonuclear leukocyte. Oh, I think the oldest might have 5 or 6 lobes, and they generally live, I think, for no more than about 21 days. Some of these granulocytes will live as short as 7 to 12 days.
What does their, how does their reproduction What's the point?
Yeah, I don't really know the point of having these different lobes. I really don't. It's the same nucleus, it just divides into different lobes. Because your mature neutrophil in the circulating peripheral circulation, it's not undergoing cell division.
Yeah.
That's it. So,
That way because it's that way, there's not really a reason you're saying?
not that I'm aware of. Not that I'm aware of. It's just the age of the neutrophil can be estimated by the number of lobes. When they're new, they're fewer. When they're old, they're more.
Like rings on a tree.
Somewhat. Except this thing's not really growing. Now, the important part about neutrophils is they are going to be the most abundant of your white blood cells, and of course then the most abundant of the granulocytes. You may have as many as 70% of your white blood cells present as neutrophils. Now, when you have that many neutrophils in the bloodstream, is it any surprise that the neutrophils are going to be the very first to arrive at the scene of the infection? Yeah, if you want to completely eliminate crime on the planet, if you want to eliminate speeding on the highways, have a trooper every mile, right? Well, you got a neutrophil at every scene of any attack that could happen, and so they're ready to take it on immediately. And what do they do when they encounter a pathogen? They eat it. They gobble it up. Get it out of the way, prevent it from reproducing, take it out of the circulation, dispose of it.
So do neutrophils have more like lysosomes and peroxisomes?
I wouldn't say more, but all these granules have those specific enzymes in them as well as chemokines to attract other immune system cells into the area. But they're going to have a lot of degradative enzymes because they are phagocytic. So they're going to eat it, break it down, and gobble it up. Now, this type of granulocyte, you can already see that the granules are much larger than we saw with the neutrophils. And in fact, histologists, when they stain blood preps, blood smears, with a stain called eosin, which is, it stains material red, these granules really picked up a lot of these red granules. So they said, oh, these granules love eosin. They love red. So that's how we got the eosinophil, because of those deep red granules that almost obliterate your ability to see the bilobe nucleus. These are really only ever going to have 2 lobes to their nucleus. They're much less abundant than we see with the neutrophils. And these, more than just undergoing phagocytosis of anything and everything, they're really going to be focused on attacking multicellular parasites. Now, one of the first things you're going to do when a patient comes in with an infection is a CBC. You're going to be doing blood work. What's going on? And you're going to be looking at the differential count of your white blood cells. And if you see an astronomical amount of eosinophils, that's going to tell you there's an infection. Start looking for the parasite. So that's one way these can be diagnostic. When these cells encounter a parasite, they can't engulf it. You saw the tapeworms, right? Yeah. So they are going to throw bombs at the parasites. They're going to expel these cytotoxic chemicals to try to destroy the parasite and recruit other immune system cells into the area to help remove the pathogen.
So are these chemicals almost always like local and specific to?
Yeah, they're going to have to be local. That's why you're going to have to get more help coming into the air, because one of those is not going to take care of a 12-foot tapeworm.
What's to stop a healthy cell from taking that cytotoxic chemical and it dying?
Again, a lot of times you're going to encounter these parasites in your interstitial spaces. You don't necessarily have a lot of cells, but depending on what the parasite is, you may have to kill a few good cells to get rid of the one bad. It's like cancer. You can cure cancer, but you just about have to kill the patient to do it at times. So it's a trade-off. Now, I'm sorry.
Are these like ants on something? Like, would they just completely surround it to kill it, or would they just float by and like?
Well, they're going to be recruited into that area, and it's kind of like what Garrett was talking about. If you're going to be releasing toxic chemicals You don't want it to be too widespread. You want it to just affect that parasite. So you're going to want to keep it as close to the parasite as possible. So our last type of granulocyte are the basophils. And when we talk about acidic and basic, acidic is usually red in color. And what color is usually basic? Blue. And so the granules of basophils are the largest of the granules. They're deep, deep blue. And they basically, that's all you're going to see are these granules. It's going to be very difficult for you to even see the nucleus. And this is often a bilobe nucleus as well. But it's these big, dark blue granules that are going to be the characteristic of what you're going to see with the basophils. Now, basophils, that's their name when they're in the blood vessel. But when these undergo diapedesis and crawl out into your interstitial spaces, they change their name. Basophils then are called mast cells. Another name that people will often use for mast cells is tissue basophil. So either a tissue basophil or a mast cell, I'm good with either one of those. But I want you to understand we're talking about the same cell, just in a different location. Now, of the granulocytes, this is the most rare, which is probably a good thing. You've only got 0.5% of all white blood cells as well when you look at all leukocytes, including lymphocytes. But it's good they're rare because these are going to be involved in inflammation and allergies. Does anybody have some really, really bad allergies? You got your EpiPens handy and stuff? Guess who you have to thank for that? These guys, the basophils. And it's because of the chemicals that they secrete. And we're going to touch on these more in a bit. But the basophils, mast cells, depending on where they are, these granules are loaded up with histamine as one example. Histamine is going to cause your blood vessels to dilate. Now, why would you want your blood vessels to dilate if you've got an infection? You got more blood coming through, which means you got more white blood cells coming to the area. You're recruiting additional help. Increasing capillary permeability. You want these cells to be able to get out of the bloodstream and get to where the infection is quicker. But if you make the blood vessel more permeable, and now you are exposing some of these proteins outside of the capillary to a lot of the plasma proteins, to the platelets, what could that lead to? If you cut your vessel, what's going to start happening? Platelet plug, coagulation, forming a blood clot. So in these granules also, you have an anticoagulant. You want it to be leaky to get those white blood cells, those leukocytes, to the infection, but you can't start clotting or you've defeated the purpose. You're never going to get additional cells into that area. Now, do we ever have problems with histamine? In clinical settings, are you ever going to have patients that have a problem with histamine? Do you ever have problems with histamine? Yes, you do. When you have a stuffy, runny nose, what do you go pull out of your medicine cabinet? An antihistamine. Because when your blood vessels are more permeable, you're having inflammation and congestion in your nasal area. It's all because of these cells that are making you feel that way. Yeah.
So if basophils are the main ones responsible for allergy responses, what role do the eosinophils help them?
How will the eosinophils help them?
Well, yeah, just because like in our lab textbook, it does say also that the eosinophils help with allergy and asthma.
Depending on what is causing it, basophils, especially with really, really severe allergies, you have to have a means with which you turn it off. So my guess in that particular case is you're going to be able to secrete some of those cytotoxic chemicals from eosinophils, and that might help mute a really, really expansive basophil mast cell allergic reaction.
Right.
Because if you don't turn that thing off, if you don't have your EpiPen, it's not going to end well. So you got to have a way to shut that down if it's out of control. That's kind of my guess with the eosinophils. I've never heard of a real good connection in my head, what makes sense. All right, now we're moving to the next group. See the A? What does that mean? No granules. And so the largest of our white blood cells in the peripheral circulation, you see these are red blood cells. Remember, about 8 microns in diameter? These things are huge, but you don't see the granules, but you do see this really large, either horseshoe-shaped, some will say indented nucleus. And these are going to be monocytes. They don't last as long in the bloodstream, maybe 1 to 3 days. And while neutrophils are the first at the scene of the crime, monocytes are going to be last to get there. Now, much like basophils, as soon as the monocyte undergoes diapedesis and gets out, we change the name to a macrophage. So basophils become what when they leave the circulation? Mast cell. Monocytes become macrophages. Now, I don't understand how this all came about, but you know, when scientists discover things, they like to name things after themselves. So there are a lot of different macrophages in the body in different places that have different names. In your epithelium, you have these macrophages that have these long processes, but they're still macrophages. But somebody said, you know what, let's call these dendritic cells. Dendrites, dendrology is the study of trees, so you see all these branches, all right? So that's one other name for macrophages. In the lungs, call them dust cells. In the liver, Kupffer cells. Dr. Kupffer said, oh, I want that name. So macrophages are practically ubiquitous cell types. But they can have different names, but understand all of these do the same thing. Regardless if they have maybe a little different shape, if they have a different name, if they're in a different location. So these definitely are phagocytic cells. They're going to gobble up material. But the other thing that's going to be important as we move through these different functional divisions of the immune system is these cells, the macrophages, the monocytes, dendritics, Kupffers, dust cells, they are also going to function as antigen-presenting cells. Do you remember us talking about those when we talked about the T cells in the thymus and how they get screened? T cells cannot respond directly to an antigen. They have to have help. They have to have someone show them and display it to them. And we'll get into that dynamic of antigen presentation probably on Thursday. But these are a big group of antigen presenting cells that are going to be able to activate your cell-mediated immunity. And to a lesser degree, our B lymphocytes, and we'll get into that in a little bit. Now we're still with the A granulocytes. No granules. We left those with the neutrophils, eosinophils, and the basophils. But now we're in our lymphocytic lineage, not the myeloid lineage anymore. And we have 3 types of lymphocytes, the Bs, the Ts, and the natural killers. You can also call these the cytotoxic T cells. Now, where did the T come from here for T lymphocytes? Thymus. That's where they're screened. That's where the T lymphocytes mature. And if everything works right, they get to leave the thymus and go to other secondary lymphoid organs. Here's a trick question. Where does the B come from in B lymphocytes? I mean, that's a B. So originally, now in our bodies, it's bone. But that's just an absolute coincidence because when B lymphocytes got their name, they were discovered in birds, and the B lymphocytes mature in an avian organ called the bursa. We don't have a bursa, but we're just lucky that it's still bone. So B lymphocytes mature in our bone marrow, T lymphocytes mature in the thymus. And the natural killers, those are, I believe they're also going to be in the thymus. I need to look that one up. I think they're also thymus.
Do B lymphocytes also need antigen-presenting cells?
They can, but they don't have to have them.
Okay.
Because these guys are going to have special receptors on their cell surface that are basically going to be the antibodies that they secrete. And if something binds to that antibody on their cell surface, then you know it's a match and they can start making more. Yeah. Yeah.
I'm assuming our versus around our joints are not the same as the.
No. Those joint bursts, yeah, completely different thing. Now, these cytotoxic Ts, these are going to destroy your own body cells if they become tumorigenic or if they're infected with viruses. That's why we say defective somatic cells. And it's going to be these chemicals. I mean, the killer Ts, the cytotoxics, they're not phagocytic. They release hand grenades and bombs that are going to kill these defective cells. Again, the killer T lymphocytes are also going to be able to destroy bacteria with those chemicals.
So the rule says that it's mainly in bone marrow for natural killers, but
Yeah.
they've also found more researchers.
Oh, gotcha.
Secondary lymphoid tissue.
Yeah.
Results.
They're going to have some similar receptors that you just want to make sure these are not going to recognize self. So I wasn't sure where they mature, but it's mostly bone marrow? Yeah. Yes, ma'am.
All the lymphocytes destroy defective somatic cells, or is that just the?
That's the killer Ts, the natural killers, the cytotoxics. They're the ones that are going to kill those somatic cells. Yeah. Now, the Bs and the Ts, you're going to find those in every lymphatic organ, every secondary lymphatic organ. And this is where, when they become activated, they're going to help with the adaptive or learned immune response. We're going to get to these two big scale immune responses. Innate, which is going to recognize anything and do it quickly, and the adaptive, which you're going to have to learn and you're going to have a memory for over time. So these really are going to be part of our adaptive immune system. Up there we already said cytotoxic Ts. You're going to find them also lymphatic organs. You're going to find them in blood. And as I mentioned before, virally infected cells, tumorigenic cells, defective cells are going to be destroyed by these that secrete these cytotoxic chemicals. Yeah.
You said in the previous slide that they manage the adaptive immune response.
They're not the regulators of it solely,
Okay.
but they are going to help. And there's several different cells that we're not going to necessarily get into. There are also regulatory T cells that can stop a cell-mediated response. So in a way, they are going to play a role in the balance of activation and suppressing.
Okay.
All right, so we know the bad guys. Now we know the good guys that are fighting for us. Now let's see how all of your body is working together to protect you. And so we've divided the immune system up into the innate immune system, and then we mentioned the adaptive immune system, and you can see here the T lymphocytes and the B lymphocytes. We're going to cover innate first, because there's a lot of parts of your body that you may not necessarily think of as immune system, but they are in fact protecting your body from pathogens. So we're going to cover this first. Innate, you're born with it. It doesn't have to develop. They don't have to mature like your adaptive immune system. Nonspecific, it doesn't matter what it is. You don't have to have seen it before. You don't have to have a vaccine for it. Your body is going to protect you from this particular pathogen or any particular pathogen. This doesn't take any time. It's an immediate response. The best way to fight off a pathogen is keep it from infecting your body in the first place. It's kind of like, how do you cure cancer? You find it quickly before it spreads. So that's kind of like the innate immune system. The adaptive, as we've already said, it's going to involve your B and T cells, and it's acquired, it's learned. You have to be previously exposed before this is really going to be activated. But the other thing this has for those specific pathogens, the adaptive immune system remembers. It remembers that pathogen, and when you encounter it again, it's going to give an even more robust response to eliminate that pathogen. And it doesn't take a bunch of cells. One cell is going to respond to one specific antigen. And when that one cell responds, it's going to start cloning itself and making more and more and more. It's like the Clone Wars in Star Wars. I got a Star Wars reference in. You know, you're making a lot of the same army, a lot of the same soldier to do the fighting against that one specific pathogen. But innate happens instantly. This can take several days. Your first exposure is going to take weeks. The second exposure may be a day or two, but it's not going to be as rapid as you see with the innate immune system. And there we've already mentioned the memory, and we'll see how those memory cells are critical. And actually, memory cells, that's what vaccines are trying to build, are your memory cells. Vaccines aren't necessarily trying to cure you from your first exposure. All right, so innate immune system, your first line defense. Any surprise whatsoever that your skin is a first line defense against pathogens? How often do you get a skin infection unless you cut your skin? You don't. But if you get a cut and you don't clean that cut and you don't take care of that cut, what's going to likely happen? It's going to get infected. And so your skin is a physical, but we're also going to see somewhat of a chemical barrier to infection. And one of the reasons is your skin cells are tightly packed together. Do you remember the word we used? Contiguous? Contiguous all together. There's no way for pathogens to get in unless you compromise the continuity of your skin, maybe with a cut or a break.
Also the story about the airplanes.
The airplanes.
Remember how the holes in the airplanes?
Oh, right. Yeah. We don't want holes. The other thing happens is your skin is dividing at the base, and your skin is continually pushing new cells upward, 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 upward. And what happens to the top layer of skin? These are just dead cells. You shed those cells. So basically, if a pathogen is trying to burrow down in your skin, it's like trying to walk up the down escalator or trying to swim upstream. So just the growth of the skin itself can slow down a pathogen. But your skin also produces an acid, hyaluronic acid. The pH of the skin is going to be an important barrier and basically a detriment to pathogens. And we're going to see how that works. But hyaluronic acid, microbes aren't going to like this. Microbes have a very specific pH range that they feel comfortable in, and the pathogens you encounter on your skin typically aren't going to like that surface with hyaluronic acid. And so the pathogens are going to be killed by that. In fact, when you look at skin pH, this is a little surprising. What is our physiological neutral of our plasma and our tissues? 7.35, 7.4. Our skin is 5.5. Man, we're down here in the pretty acidic range. And it's really important we balance that pH, because if your skin gets too alkaline, what's another word for alkaline? Basic. So we're moving upwards. So if we get too alkaline, your skin's going to start to have issues. Flaky, red, itchy is going to be another one. Too acidic, and you might end up with eczema, and that's a problem for acne as well.
Okay, so alkaline pH doesn't incur eczema or acne.
No, it's more the acidic pH.
Okay.
And so,
what conditions in alkaline?
I'm sorry?
What kind of conditions like in alkaline?
You talking about how high would you want to go? I mean, I wouldn't want to go much more than this weak alkaline. You don't want to get in strong alkaline, and you don't want to get down here in the strong acidic. You know, we're talking about hydrochloric acid down here. We're talking about bleach up here. Yeah, don't do skincare with bleach. Not a good idea. You'll disinfect it, but not good. Here's another thing. With our 5.5 pH and the other materials and chemicals we have associated with the skin, this is going to be really hospitable for some bacteria and microorganisms to live on your skin that are beneficial. Now, all of these microorganisms we call microflora on your skin, but these particular organisms are non-pathogenic. They're beneficial to you, but really, they're not getting any benefit. A symbiotic relationship is where both organisms benefit. A commensal relationship is where only one benefits. And with the bacteria living on your skin, they're preventing other microorganisms from coming into the area because it's their turf. And so they benefit you, but they really just can live and grow anywhere. So, I mean, it's important for these to be on your skin. Just like when we talk about GI tract, you're going to have flora in your GI tract that you want there to help protect you. Hugh, did you have a question?
Yes, sir. So these microflora are able to stay on our skin despite them shedding.
Yes.
How are they able to stay if they're like trying to swim upstream all the time?
Well, again, they're not trying to burrow down into the skin. They're just on the surface.
Okay.
So when one cell starts to shed off, and just think of the numbers and the amount of these microbes on the surface of the skin, they just fill in that space.
Okay.
Yeah. Now, if the epithelium wasn't enough and the conditions of the epithelium, we're going to see that our glands help keep the skin in the right conditions. Sweat glands are going to have microbial, antimicrobial substances. And then our sebum, you remember sebum from the sebaceous glands? They're going to produce material, and that lowers the pH of the skin. It's going to keep it in the acidic range, again, making it inhospitable for pathogenic microbes. You didn't realize your skin was that big a part of the immune system. I certainly didn't. I just thought you had to use soap. I don't know. Now, cutting your skin, we know that that is definitely a way to get an infection. You break that physical barrier. But there are other ways to gain access into your body. And certainly your mucous membranes, your mouth. Because as we said, kids early in life, everything's going to go in the mouth. How often do you touch your face and your mouth throughout the day? Did we realize how much we did that before COVID? After COVID, it was like, oh, don't touch, don't touch your face, and you realize how often you do that. Well, in your mucous membranes, especially around your mouth and your oral cavity, again, we're going to have a contiguous layer. And this is going to include your GI tract, respiratory, urinary, reproductive tract, the linings of your body. And they're going to be that physical barrier. But like the skin, you're going to have chemicals. It's a biological barrier. You're going to have other secretions that are going to help with that. In particular, what's happening in your mouth, which we'll get to next. I thought that was my next slide. Respiratory tract. You can either ingest something or you can inhale material. And your respiratory tract is going to protect you from getting stuff too far down inside your body. First, you got your nose hairs. All right, physical barrier, that's going to prevent particles from getting anywhere past your nasal passages there. But if you do get further and you get down into your the bronchi and the bronchioles of your lungs, you're going to encounter this kind of epithelium. Do you remember what kind of epithelium this is from your histology on epithelium? I think I heard it's columnar. And we know that we've got cilia on the surface. But this epithelium is specific for respiratory epithelium, and it's not stratified. Pseudostratified columnar epithelium that is ciliated on the surface. Now, do you see these sort of oval-shaped dudes right here? Did y'all have to identify those? These are called goblet cells. And goblet cells make and secrete mucin, which is an important part of mucus. And so these goblet cells produce mucus all along the surface of the cilia. So if pathogens get past your nose hairs, if they get down into your airways, they get stuck in this sticky, thick mucus on the surface, and they can't escape. I don't know about you, but I don't want nasty garbage-filled mucus just lining my airway, right? Well, it doesn't stay there because these cilia move. And these cilia beat in such a way that this mucus that now contains all the nasty stuff is moved up in your airway until you get to your pharynx, your larynx, and then you either hork it up or you swallow it down. Now, getting it out of your body, you think, well, that's the preferred way to get rid of the pathogen. But when you swallow the pathogen, what's the first place that pathogen is going to take a swim in? Hydrochloric acid. And we're going to get how the GI tract does that in a bit. Did I see a hand? Somebody have a hand? Yeah.
What's the pharynx exactly again? I know it's in the throat, right?
This is going to be the pharynx, larynx in this region where you have the epiglottis.
Okay.
Because you have the esophagus and the glottis, which is the opening of the trachea, kind of in the same place.
Yes, sir.
And once you get that material up to the glottis, now you can swallow, goes around the epiglottis and into the esophagus into your stomach.
Okay, or spit it out.
Or spit it out.
Okay.
Now, coughing, sneezing, I love this, blows microbes away from the lungs. Yeah, it blows microbes everywhere, right? Sneeze into the bend of your arm. Didn't we learn this during COVID? Sneeze into your sleeve, have something you sneeze into, don't just blow it in your friend's face. Yeah. Now, here's our GI tract. Now we talked about the hydrochloric acid in the stomach. We're going to get to that. But you have antimicrobial chemicals in the saliva in your mouth because you don't want bacteria growing in your mouth, right? A lot of the bacteria can cause cavities on your teeth in the accumulated material. That's why you need to brush your teeth. You also have anticoagulants in your saliva because when you eat and you chew, probably when you're eating the chips and salsa. Have you ever cut your gums on the chips and you bleed? Well, every time you eat, you're going to cause micro breaks in your mucous membrane and you don't want to bleed. So you got a lot of really good chemicals and materials in your saliva to protect you and also to protect you against microbes. There's hydrochloric acid. Anything that gets into the stomach, it's going to take a bath in pH 2 material. Now, do you ever wonder how you can have hydrochloric? You see the movie Aliens, right? Acid for blood. How can you have pH 2 acid in your body and it just doesn't eat through your body? The cells that line your stomach produce a mucus. It's a coating mucus that is filled with bicarbonate, which buffers the acid at that lining interface so you don't basically digest yourself. Now, if you damage the stomach lining and you damage those cells that make that mucus and now the acid starts to eat a hole in your stomach, what problem do you have? Sorry? Ulcers. You have an ulcer, peptic ulcers. That's going to be in the lower part of your stomach. So hydrochloric acid, yeah, it's going to destroy everything. Now, if you have problems in other areas or how massive this problem is, if you've got some major problems in your stomach with pathogens, you're going to get rid of it, right? You're going to reverse old peristalsis and get it out. And in your intestines, if you've got pathogens and your intestines know we don't want this here, you are going to reverse the water hose. You know what I'm talking about, right? Yeah. Your intestines are not going to absorb water. They're going to excrete water, and your feces is going to be somewhat watery. Do we understand each other? Diarrhea. And if you have intestinal flu, it's like massive diarrhea. But it's flushing, trying to flush these pathogens out of your intestinal tract. Are you writing that down?
Got it.
Now, just like on your skin you have that commensal microflora, so you have in your stomach as well as in your intestines. Those are important to keep that balance. That's why sometimes if you have a bacterial infection and you have to take a pretty high dose antibiotic, sometimes your digestive system is not going to play nicely. That's because those antibiotics are killing the good bacteria, and it's going to take them a little while to repopulate, and you'll feel better. Your genital tract, all these different ways that pathogens can gain access. Yeah, you can follow the urinary system, but your urine is going to flush that out. And you also have cells that produce lactic acid, much like we saw with hyaluronic acid on your skin. And this lactic acid is going to be secreted to try to prevent infection in the genital tract, especially of females. Because we know pathogens, bacteria, and particularly yeast, like warm, moist regions, right? So you're trying to protect your body against these kind of infections. So lactic acid lowers the pH, prevents or at least slows the growth of microbes, bacteria, pH, and prevent those infections. Yeah?
2 questions. The commensal microflora, is that also not a symbiotic relationship
Correct.
in the gut?
Yep.
So what is
It seems like it would be symbiotic to me.
right, because
but-
it's eating whatever your host eats.
I mean, we're
right?
just giving them a home. I mean, to me, that's beneficial.
Okay, interesting.
But I guess they could live in anybody's GI tract. But
And then is lactate acid the same thing as lactic acid that
yes.
you experience?
Lactate is another name for lactic acid.
Okay.
So we've come through the skin and some of these non-specific defenses. Now we're going to get down to where we're getting a little bit more detail with the cells. We've already named them, and the chemicals and some of the responses that we have. We're still in innate immunity. We're not recognizing specifically what the pathogen is. We just know it doesn't belong there, and we're going to get rid of it. So when we look at the cells, and we've named all these, cells recognize these pathogens because we're looking for signals, these foreign proteins or carbohydrates on the surface of those pathogens in the coating that is called the glycocalyx, or this is sugar, glucose. Glycocalyx is the sugar coat that contains proteins. And that's how these cells of the innate immune system are going to know, you don't belong here, we got to get rid of you. Now we also have the immune cells that are going to have certain receptors, and this is going to be more of the innate immune system that we'll get to in a bit. But this is pattern recognition of these receptors with antigen presented on them, and we'll get to that when we get to the adaptive immune system. Yeah, if you don't have the right match or you're not getting the right signals, then you don't belong. And we'll have you drink the Kool-Aid or whatever.
Glycocalyx is a sugar coating?
So when, again, early histologists looked at cells, they saw that the outside surface of many cells looked fuzzy. And they said, oh, it's got a cell coat. Well, when they started analyzing what was the coat made of, they found a lot of proteins and they found a lot of carbohydrates. And so they just named this early on the glycocalyx, and that name is kind of perpetuated, even though we know in a lot more detail what the material is on those cells.
So glycocalyx is on our own innate immune system?
Yeah, we have glycocalyx. A lot of the bacteria, they have glycocalyx, and the single and multicellular. Well, I'm not sure about the single-celled eukaryotic parasites, but the multicellulars do.
So it's both on our cells and the antigen?
Carbohydrates. The glyco is short for glucose, sugar. So many of our leukocytes, other than the lymphocytes, many of them are going to be phagocytic in nature, and principally the neutrophils and the macrophages, or any other name that we want to use for those particular cells. And this is the cell eating, right? Engulfing that material, kind of like an amoeba. You engulf it, you process it, and you spit out the waste. But what happens when neutrophils and to a degree macrophages, when they start eating and phagocytosing all of these pathogens, they get so bloated and they get to a fill point that then the cell kills itself. It undergoes apoptosis. And that then, dead and dying neutrophils and macrophages are what results in this lovely thing called pus. I have looked up, is there a scientific name for pus, and I can't find one. Pus is just pus. It's one of those words that your face tells you what the word is, right? Because you never see someone go, pus. Smiling. No, it's always pus.
You know those YouTube
Pus.
pimple poppers?
Huh? No, I'm not. I ain't watching that. No. Now, dendritic cells, and remember, this is in our epithelium, like in our skin. And again, the other monocytes, macrophages, we already said they're going to be antigen-presenting cells. Oh, you found a name? Okay, that's just French. Okay. So, antigen-presenting cells. These cells are going to activate the adaptive immunity, especially for our T lymphocytes and to a lesser degree our Bs. And so I want you to remember macrophages and all those other named macrophages are antigen-presenting cells. Got to have them to activate the T cells. Here we're going to, eosinophils, phagocytic to agree, but these granulocytes, especially the eosinophils and majorly so the basophils, when they encounter a pathogen, they undergo a process called degranulation. Because again, as these early histologists were looking in microscopes at these granulocytes, they would see the granules. But then when they would add certain chemicals or they would add certain pathogens onto the slide with these granulocytes, then they would look, and guess what? The granules were gone. How did this cell degranulate? Well, it shot all the granules outside of it. It released all those chemicals and the granules were just gone. And so again, we mentioned with the eosinophils and the multicellular parasites, you're going to launch all of these toxic chemicals out of the eosinophil in the neighborhood of that parasite. We've got a nice little roundworm looking thing here. And that is going to kill that particular parasite. Basophils, they also undergo degranulation. In the case of allergies, this is going to coincide with antibodies. And this degranulation is going to happen massively. It is going to happen rapidly. And this is when you go into anaphylaxis and better find the EpiPen. So when basophils, mast cells, undergo massive degranulation, that's when you can get in trouble.
Is your innate immunity something you inherit from your parents?
Innate?
Innate immunity, yes.
Well, innate immunity is like what we talked about before. This is your skin, this is your GI tract. So it's something we're born with and we all have it. The adaptive immune system, I would think, is more of what you're genetically going to inherit from your parents.
Okay.
So what, like in the eosinophil itself, allows it to have cytotoxic chemicals inside of it but still not like die?
Well, again, these chemicals are all in vacuoles. I mean, membrane-bound vesicles inside that cell. And when it releases, then it's out there to do its thing. It doesn't do it inside the cell. It's like lysosomes inside the cell. Those are digestive enzymes, but it doesn't digest the whole cell. It just digests what's trapped inside that vacuole.
So the toxins that come out of the vacuoles are what cause people to go into anaphylactic shock?
No. For the cytotoxic chemicals, they're attacking the parasite. For basophils, the heparin, the histamine that causes your blood vessels to get leaky, if you have every blood vessel in your body get leaky at the same time, your blood pressure is going to plummet. And that's going to be one of the signs of anaphylaxis. You could lose consciousness.
Well, can the cytotoxins in
Eosinophils.
yep. Can that like hurt us?
Well, remember, you've got vastly fewer of the basophils than the eosinophils. So the eosinophils don't have that allergic response type situation, whereas the basophils do. So no, the eosinophils aren't going to get to that point that they're going to be so detrimental. All right, basophils, mast cells. Basophils, mast cells are really functioning with inflammation and signaling and facilitating inflammation. Now remember, basophils are in the blood. What do we call them in the tissues? Mast cells. Mast cells. And mast cells are everywhere. Your interstitial spaces, mucosa, internal organs. The basophils are just in your circulation. And you can see in this illustration this particular basophil, which, again, this is an artist picture, right? Because a basophil is just going to be a little bit bigger than a red blood cell. And this is a capillary. How big is a capillary? 1 red blood cell in diameter. So this is like King Kong of cells. Yeah. So what they're illustrating is how this cell, and outside the bloodstream, this will be a mast, not a basophil, but how it's responding to certain chemical signals and secreting histamine, heparin, and this new one that we have, which is called eicosanoids. You remember eicosanoids? Talked about them as bioactive agents. The eicosanoids, that's going to help increase fluid movement from the blood into the tissue. This is where you're going to begin to flush the tissue more, producing more lymph, but you're also allowing more chemotactic signals to get from the tissue back into the blood to recruit more cells. Everything you can do to recruit more of the leukocytes, the white blood cells to the site of the infection, is going to be most beneficial. Natural killers, cytotoxic T cells, excuse me. Again, unwanted cells, I don't really like that term. Your book used that term. These are defective cells. And I guess defective cells are unwanted, right? If they're tumorigenic, if they're virally transformed. And so these cytotoxic T cells, they're kind of the patrollers of the body. They go around screening, detecting cells. The transplanted tissue, we said, if you don't have a close enough match, these are the cells that will lead to a tissue rejection. And the way they accomplish that is, first, these cytotoxic T cells, they secrete materials called perforin. Perforate. What does it mean to perforate something? Poke a bunch of holes in it. Well, these proteins will basically make a transmembrane hole in the bad cell. And so everything leaks out. Then you secrete these granzymes, and these again are digestive enzymes and chemical signals that cause the cell with a bunch of holes in it now to undergo apoptosis. So you can see here's the cytotoxic T cell attacking the unhealthy cell, the defective cell, and it ends up you just have this waste product out here, and that's great food for macrophages to come in and clean it up, and now you've just eliminated the situation. Now we also have chemicals. We talked about the chemicals in those granules of neutrophils, eosinophils, and basophils. Maybe not digestive enzyme, but these are signaling molecules. They're called cytokines. And one of those particular cytokines is called interferon. And so in this case, cells can secrete interferons. And the name interferon comes with a process that this cytokine will interfere with replication of an infected cell, particularly viral replication. So you interfere with replication in a defective cell, you interfere with replication in a virally transformed cell. That's going to slow that cell down. Now the killer T can come in and finish it off.
So, aren't doctors like able to use like pharmacological interferon?
So this was years and years ago with various chemotherapeutic agents for cancer. Someone discovered somewhere, and I'm not sure how prevalent it is today, but discovered that if you augmented interferon with a certain chemotherapeutic, that it enhanced the benefit. It killed some of the cancers faster. I don't know if there's a specific type of cancer that responds more favorably to the interferon treatment or not. But again, interferons are typically seen to deal more with virally infected and virally transformed cells. Now, as long as we're talking about chemicals, this is a big group, and this is the last thing I want to cover today. But when we look at this group of proteins, first of all, I want you to understand that these proteins are present in the plasma of your blood already. But these proteins, and maybe 30 of them, they are inactive. They have to be activated. You remember the clotting cascade? You have these inactive, these OGen proteins that are present in your blood. These are very similar, and in a similar way, there are two ways that you can activate these complement proteins. And these complement proteins are going to have a wide-ranging impact and physiological consequence that's positive for the immune system. So in our illustration, first, you can activate the complement cascade, as I call it, through what's called the classical pathway. And in the classical pathway here on the left, you see an antibody has recognized an antigen. That then is going to recognize a complement complex of proteins that is going to first activate C1. Now, you don't have to remember all these numbers and the sequences, all right? That's not important to me at this point. But when you have an antibody and a pathogen, that comes in contact with the first complement protein, that's going to activate. That then becomes an enzyme that activates the next, that activates the next, and activates the next. And you break these complements into subunits, and typically you'll see them as A subunits and B subunits. And so as you go through this cascade, that is going to lead to the activation of the complement proteins and downstream immunological and physiological effects. You can also start this process in what's called an alternate pathway when you recognize a foreign sugar, polysaccharide. Saccharide is sugar, right? So when you recognize a foreign sugar on a bacteria or other pathogen, that's going to activate a different complement protein that's going to follow a pathway that leads to a common point that then can activate these downstream physiological and immunological responses. So you can start it one of two ways, but you're going to end up in the same place, regardless of which way you start it. And the downstream processes are opsonization, coating the pathogen with antibodies, inflammation that we kind of talked about a little bit already, or cytolysis, which basically cytocell lysis means what? To break. So breaking the cell, killing the cell. So opsonization first from our previous, I blew this up a little bit. You have these complement fragments, and they have now decorated that pathogen. Much like you can have opsonization with antibodies, when antibodies decorate a pathogen, it makes it irresistible to phagocytic cells. So here with complement, since complement starts with a C, this opsonization, I call it chocolatification. Because if you put chocolate on anything, I'm going to eat it up. Well, if you put these complement fragments on that pathogen, these phagocytic cells cannot resist it. And they eliminate it very, very quickly. Now we can see other fragments of other complement proteins are going to influence other immune cells, in this case basophils and mast cells. Once you activate the basophil mast cell, it's going to degranulate. And what are the two big bioactive chemicals that are going to be released from a mast cell or a basophil? Histamine and heparin. That is going to lead to inflammation. And then lastly, we have a fragment that's going to activate these other sequence of complement proteins. These complement proteins are going to come together and they're going to make a barrel, a protein barrel that's going to insert itself into the plasma membrane of the pathogen, the bacteria. So basically, you're going to make a protein hole in the pathogen, and all of the stuff is going to leak out and fluid's going to leak in. Anyway, it's going to be a bad day for that pathogen. And that then kills the pathogen, the infected cell, and it's going to get rid of it. I am always amazed when we go through the immune system, we talk about all these different mechanisms. Why do we ever get sick? Because there's so many pathogens out there. And there's so many things that can go wrong with your DNA and mutations and everything else.